You have probably heard that tirzepatide outperforms semaglutide in clinical trials. The SURMOUNT-5 data is unambiguous. 20.2% average weight loss versus 13.7% at 72 weeks. That gap is real and it is clinically significant. But the number is not the interesting part. The interesting part is why. What is happening at the molecular level inside your body that produces an outcome a single receptor drug cannot replicate? What does it actually mean to be a dual agonist? Why does hitting two receptors instead of one change the results so dramatically? And here is the question that should genuinely unsettle anyone who thinks they already understand this drug. How is it possible that stimulating the GIP receptor and blocking the GIP receptor can both produce weight loss? That is not a simple question, and the answer reveals something genuinely fascinating about how tirzepatide works, something the headlines and the clinical trial summaries never get around to explaining. That is what today is about. Let's start with the structure. Because to understand why tirzepatide behaves the way it does, you need to understand what it is built from and how it was designed. Tirzepatide is a 39 amino acid peptide. If you have been following our other videos, you know what that means. A short chain of amino acids, small enough to carry a targeted biological instruction. 39 is on the longer end for a therapeutic peptide, but that length is part of what allows it to do what it does. Now, here is the first thing that surprises most people about this molecule. Its backbone, the base sequence it is built from, is actually modeled on GIP, not GLP-1. The engineers started with the natural GIP structure and then grafted GLP-1 activity onto it. So what you end up with is a molecule that is primarily a GIP agonist. It fits the GIP receptor more naturally, with enough GLP-1 character built in to activate that receptor too. It is not a 50-50 split. It never was. More on that later, but keep that asymmetry in mind because it is essential to understanding why this drug behaves the way it does. Now, the molecule as it comes from nature would not survive long enough to be a useful drug. Natural incretin hormones like GLP-1 and GIP are cleared from the bloodstream within minutes. A therapeutic that vanishes before it can act is not a therapeutic. So the design team made a series of targeted modifications, not one massive edit, a collection of small, specific tweaks, each one serving a particular purpose. At several positions along the chain, the natural amino acid was swapped out for a modified version called AIB, alpha-amino-isobutyric acid. Each of those swap points makes the molecule slightly more resistant to the enzymes that would normally break it down. At the tail end of the molecule, what biochemists call the C-terminus, there is an amide cap, another small structural modification, another layer of stability. And then there is the most important modification of all. At position 20 of the amino acid chain, there is a lysine residue. And hanging off that lysine is a long fatty diacid chain, a carbon-20 fatty diacid to be specific. That fatty diacid tail is what makes a once-weekly injection possible. Here is how. Your bloodstream is full of a protein called albumin, the most abundant protein in your blood plasma. Albumin is essentially a transport vehicle, constantly circulating, carrying various molecules around the body. The fatty diacid tail on tirzepatide can grab onto albumin. Once it latches on, the drug is essentially hitching a ride on the most common protein in your circulatory system. Instead of being filtered out and excreted, it circulates along with the albumin. That single structural feature, a long fatty chain that binds to albumin, is what extends tirzepatide's half-life from minutes to approximately a week. One injection, 7 days of activity. That is the engineering behind it. Now let's talk about the two receptor systems tirzepatide activates, starting with GLP-1, because that is the one most people already have some familiarity with. GLP-1 stands for glucagon-like peptide 1. It is an incretin hormone, a signaling molecule released by cells in the lining of your small intestine in response to food. The cells that produce it are called L cells, and they sit in the gut wall waiting for nutrients to arrive. Once in circulation, native GLP-1 has roughly 2 minutes before enzymes break it down. 2 minutes to do its work before it disappears. Pharmaceutical engineering, as we just covered, has extended that window dramatically for therapeutic applications. So what does GLP-1 actually do when it activates its receptor? The first thing, and this is the effect the drug was originally developed for, is glucose-dependent insulin secretion from the pancreas. GLP-1 receptor activation tells the beta cells in the pancreas to release insulin. But here is the critical qualifier. It only does this when blood glucose is already elevated. That glucose dependence is not a minor detail. It is what makes GLP-1-based drugs fundamentally safer than older insulin secretagogues. Previous generations of diabetes drugs forced insulin release regardless of blood sugar level, which created a dangerous risk of hypoglycemia. The blood sugar could drop dangerously low even when the patient had not eaten. GLP-1 receptor activation does not do that. At low blood glucose, the incretin effect is limited. The signal backs off when it is no longer needed. The system has its own built-in safety check. On the other side of the pancreas, the alpha cells, GLP-1 signaling does something complementary. It reduces inappropriate glucagon secretion. Glucagon is the counter-hormone to insulin. Where insulin lowers blood sugar by shuttling glucose into cells, glucagon raises blood sugar by signaling the liver to release stored glucose. During fasting, that is a useful and necessary function. But in a state of elevated blood sugar, excess glucagon makes the problem worse. GLP-1 receptor signaling tells the alpha cells, not right now, suppress the glucagon release. Let the insulin do its job. Then there is the stomach. GLP-1 receptor activation slows gastric emptying, the rate at which your stomach moves its contents into the small intestine. When digestion slows, nutrients are absorbed more gradually, blood sugar rises less steeply after meals, and critically, you feel full sooner and stay full longer. And then there is the brain, and this is where the appetite side of the story lives. GLP-1 receptors are found in several regions of the brain involved in appetite and satiety. There is the vagus nerve, the communication highway between the gut and the brainstem, which carries GLP-1-related signals upward. There is the nucleus tractus solitarius in the brainstem, which integrates inputs from the gut and visceral organs. There is the area postrema, which sits outside the usual blood-brain barrier and responds directly to circulating hormones. And there is the arcuate nucleus of the hypothalamus, a critical appetite regulation region. In the arcuate nucleus, there are two key neuron populations working in opposition: POMC neurons, associated with satiety signaling, and AGRP neurons, associated with hunger signaling. GLP-1 receptor activation in this region tips the balance towards satiety. Reduced appetite, reduced food preoccupation, the internal background noise of food cravings, what researchers and patients both call food noise, gets quieter. That multi-tissue mechanism, pancreas, stomach, brainstem, hypothalamus, is why GLP-1 receptor agonism produces effects well beyond what you would predict from any single site of action. It is not one thing happening in one place. It is a coordinated signal propagating through multiple biological systems simultaneously. That is the first receptor. Now let's talk about GIP. And to do that properly, we need to acknowledge something unusual. GIP spent years being the most misunderstood molecule in incretin biology. GIP stands for glucose-dependent insulinotropic polypeptide. Like GLP-1, it is an incretin hormone released from the gut in response to food. Like GLP-1, it contributes to insulin secretion from the pancreas when blood glucose is elevated. On paper, GIP looked like it should be straightforward. In practice, the research picture was significantly murkier. GIP receptors are found in the pancreas, as expected, but they are also found in adipose tissue, your fat cells, in bone, and in the brain, specifically in hypothalamic appetite circuits. The fat tissue finding is where the confusion started. Early research framed GIP primarily through the lens of adipose biology, how it behaved in fat cells. And in that context, GIP looked like a nutrient storage signal. It appeared to promote fat uptake and storage under certain conditions. That framing created a genuine concern. If GIP tells fat cells to store more nutrients, why would you want to activate the GIP receptor in a drug targeting weight loss? That logic was compelling enough that researchers actually pursued GIP receptor blockers as potential weight loss drugs. The reasoning was, if GIP promotes fat storage, remove the signal and fat storage should decrease. Some mouse model data appeared to support this. Mice with disrupted GIP receptor function showed reduced diet-induced obesity in certain models. And then Tirzepatide arrived in clinical trials and produced dramatically better weight loss than any pure GLP-1 drug, while activating the GIP receptor, not blocking it. The field had to reconcile two things that did not seem compatible. Activating GIP produces significant weight loss. Blocking GIP also produces some weight loss. Same receptor, opposite interventions, both in the direction of less body fat. This is the part of the science that makes tirzepatide genuinely fascinating, and it requires a slight shift in how you think about receptors. Most of the time, when we talk about a receptor, we talk about it as if it is one thing with one function. Activate it and you get outcome A. Block it and you get outcome B. Clean, predictable, simple. GIP does not work that way. What the research has gradually established is that GIP receptor signaling behaves differently depending on which tissue it is occurring in. In adipose tissue, fat cells, GIP receptor activation is associated with the nutrient storage effects that generated the early concern. That part of the original hypothesis was not wrong. In the brain, specifically in hypothalamic appetite circuits, where GIP receptor-positive neurons have been identified, GIP receptor activation appears to do something quite different. In the hypothalamus, GIP receptor-positive neurons have been identified in regions involved in appetite regulation, and a subset of those neurons are GABAergic, meaning they use GABA as their signaling molecule. GABA is the primary inhibitory neurotransmitter in the central nervous system. When GABAergic neurons fire, they inhibit downstream activity. So here is the proposed mechanism. GIP receptor-positive neurons in the hypothalamus are involved in appetite regulation. Some of those neurons, when activated via the GIP receptor, release GABA, which inhibits neurons downstream in the hunger circuit. The result is reduced food intake signals coming from the brain. Preclinical studies on GIP receptor signaling have shown reductions in food intake through this pathway. The central nervous system effect, through GABAergic inhibition of appetite circuits, moves in the direction of satiety, not storage. So the picture that emerges is this. In adipose tissue, GIP receptor activation is associated with nutrient storage effects. In the brain, GIP receptor activation in appetite circuits is associated with reduced food intake. Same receptor, different tissue environment, different functional outcome. This is not a contradiction in the biology. It is a demonstration of something the field has had to increasingly accept, that receptor names do not imply uniform outcomes across tissues. The same receptor, in a different cellular environment connected to different downstream signaling networks, can produce effects that look nothing like each other. And the GIP antagonism story fits into this framework as well. Drugs that block the GIP receptor, like mazdutide and AMG 133, produce weight loss primarily through the adipose pathway by reducing the fat storage signal in peripheral tissue. Tirzepatide, by activating the GIP receptor, produces its central nervous system weight loss effects through the brain pathway. Two different tissue targets, two different mechanisms, both resulting in less body fat. There is one more layer to the GIP story, and it is one of the more counterintuitive ideas in incretin biology right now. It is called the functional antagonism hypothesis, and while it is still a hypothesis, not a proven mechanism, it offers a compelling explanation for something that would otherwise seem like a problem. Here's the issue. If GIP receptor activation in adipose tissue promotes nutrient storage, and tirzepatide is strongly activating the GIP receptor, how is there not a net fat storage effect that offsets the weight loss from the central nervous system pathway? The functional antagonism hypothesis proposes that the answer is in receptor biology itself, specifically in receptor desensitization. When a receptor is activated repeatedly and chronically, day after day, week after week, the cell does something protective. It reduces the number of that receptor on its surface. The cell pulls receptors inward, away from the membrane, in a process called receptor internalization. The result is that after prolonged chronic exposure, the tissue becomes progressively less responsive to the signal. The same dose produces a smaller and smaller effect over time. In the adipose tissue, where GIP receptor activation was driving nutrient storage effects, chronic tirzepatide administration might produce progressive receptor downregulation, gradually reducing that storage signal until the adipose tissue is functionally behaving as if the GIP receptor is being blocked rather than activated. This is the hypothesis. Over a long course of treatment, chronic GIP agonism in peripheral tissue might functionally converge toward the same outcome as direct GIP antagonism, because the receptor the agonist was activating is no longer available on the cell surface. Meanwhile, the central nervous system effects, the appetite suppression through hypothalamic GABAergic circuits, continue operating through the brain pathway, which may be less subject to the same desensitization dynamics. It is important to be precise about the status of this idea. This is a mechanistic hypothesis that helps explain the observed data, not a confirmed biological fact. The receptor dynamics in human adipose tissue under chronic tirzepatide administration are not fully characterized. The degree to which desensitization is occurring and how much it contributes to the overall outcome is still an active area of research. What it does is offer a coherent framework for reconciling the paradox, for explaining how a drug that strongly activates the GIP receptor can produce results that include reduced fat mass rather than increased fat storage. In the adipose tissue, where GIP receptor activation was driving nutrient storage effects, chronic tirzepatide administration might produce progressive receptor downregulation, gradually reducing that storage signal until the adipose tissue is functionally behaving as if the GIP receptor is being blocked rather than activated. This is the hypothesis. Over a long course of treatment, chronic GIP agonism in peripheral tissue might functionally converge toward the same outcome as direct GIP antagonism, because the receptor the agonist was activating is no longer available on the cell surface. Meanwhile, the central nervous system effects, the appetite suppression through hypothalamic GABAergic circuits, continue operating through the brain pathway, which may be less subject to the same desensitization dynamics. It is important to be precise about the status of this idea. This is a mechanistic hypothesis that helps explain the observed data, not a confirmed biological fact. The receptor dynamics in human adipose tissue under chronic tirzepatide administration are not fully characterized. The degree to which desensitization is occurring and how much it contributes to the overall outcome is still an active area of research. What it does is offer a coherent framework for reconciling the paradox, for explaining how a drug that strongly activates the GIP receptor can produce results that include reduced fat mass rather than increased fat storage.
There is one more element of the tirzepatide design that is essential to understanding why it produces the outcomes it does, and it is the one detail most summaries either get wrong or skip entirely. The activity split between the two receptors is not equal, not even close. Because the backbone of tirzepatide is built on a GIP scaffold, the molecule fits the GIP receptor more naturally and activates it more strongly. The GLP-1 activity is real and meaningful, but it represents a smaller fraction of the total receptor engagement. This asymmetry has several downstream implications. First, it means tirzepatide is not simply doing what semaglutide does, plus a little extra. It is activating two different systems at different intensities through a different primary scaffold. The pharmacological profile is genuinely distinct from GLP-1 monotherapy.
Second, it helps explain the results. The GLP-1 component contributes the well-documented pancreatic and gut and brain effects that the entire GLP-1 drug class produces. The GIP component adds a separate central nervous system appetite pathway, plus its peripheral tissue effects. The two signals are operating simultaneously, but through different receptors, different intracellular cascades, and different tissue distributions. The combination is not additive, it appears to be synergistic. And this brings us back to the number that opened this video. 20.2% versus 13.7%. Tirzepatide versus semaglutide at 72 weeks in the largest head-to-head trial ever conducted between these drugs. That is not a minor performance gap attributed to a slightly stronger version of the same mechanism. That is the signature of a drug engaging biology that semaglutide simply does not reach. A second receptor system with its own appetite pathways, its own tissue interactions, and its own contribution to the overall metabolic outcome.
Tirzepatide is not a stronger Ozempic. It is a different drug with a different primary scaffold, a different dominant receptor, and a genuinely distinct mechanistic profile that reaches biology semaglutide does not touch. The 6.5 percentage point gap in head-to-head trial results is not noise. It is the signal from a second receptor system doing work that the first receptor was never designed to do. Understanding that is the difference between knowing the headline and understanding the story. If this deep dive helped the mechanism click, share it with someone still describing Tirzepatide as just a stronger Ozempic. Subscribe to What the Health for science that goes all the way down. See you in the next one.